I'll say my name is Adam Keppel, and I'm one of the founders and the CEO of Agrology. And we're really excited to have our collaborator, Dr. Michael Schuppenhauer, here today to talk about gas flux measurement in agriculture. We've learned a ton working with him, and we think that this is an extremely powerful way to understand in real time the climate positive impacts of our agricultural practices and provide really great coaching and feedback to farmers on how they are doing a better job with regenerative agriculture or just climate positive farming. So, Dr. Michael, we're ready to kick things off when you are.
Good morning and good afternoon to those of you that are on the East Coast or somewhere in the middle of the US. So my name is Michael Schuppenhauer. I'm a principal investigator. I run the research project that is a collaboration between Arbor and Lawrence Berkeley National Lab. The project is called Rice and Grits, and the underlying question that the Department of Energy asked about 4 years now was one around sustainability of agroecosystems or sustainability of bioenergy crops or feedstocks for bioenergy production. The Department of Energy obviously is focused on energy, so their agricultural questions are mostly focused on the question around bioenergy production. The question of sustainability ties back into what sometimes is known as carbon intensity. And when we're looking at carbon intensity, then we have to also look at what are the inputs, what are the outputs that are happening. So it's a fairly complex and comprehensive data gathering exercise that the Department of Energy was asking a total of 4 teams to do field research on. The field research that we're conducting on 6 sites and with other teams that are in other parts of the US are focused on the key crops. Our team is focusing a little bit more onto esoteric crops and esoteric processes, quote unquote, in comparison to other bioenergy feedstocks, which are nonetheless very significant from a greenhouse gas potential. We are somewhat at the end of our project, although we are hoping for an extension through the end of the year so that we can catch up on a number of the data gatherings that we have not completed during COVID. But I'd like to share a number of the insights that we have gained and take these ones rather as an example, not necessarily the specific numbers that I may be sharing. So one of the key things, and there was a request from the Department of Energy under the Smart Farm program, was to set up so-called flux towers. And when we do a total balancing on an agroecosystem, we got to look at the gas emissions, which is everything that is essentially above the canopy. We got to look at what is going on above ground, which is the plants and how are they forming and what is the mass balance there. And then we got to look at what is happening at the bottom, which is like what is happening in the soil. And then the last aspect is what are the inputs and outputs in terms of ag machinery to the entire mass balance and energy balance for that matter. Just to give you a scope on that one, the carbon intensity is largely dominated by the gas emissions. There is some degree of input and output from the ag machinery, but that typically is not expected to be more than 5 to 10% of the overall impact. And then the question is around what is happening on the soil side, soil carbon, soil organic carbon sequestration, and so forth. And that is a very long-term phenomenon. And the shorter-term phenomenon is really what we're measuring on a daily and then summed up seasonal basis. Flux tower sites provide an incredible insight, but it is significant capex and opex. Our stations are roughly about $250,000 per piece, and you need good people in order to maintain them. The real-time values, however, that we gather, if they are reported in real time, can enable real-time climate smart action. And we see that even in our demonstration projects, essentially, once we're drawing a conclusion out of that, farmers do actually react to that on a season-to-season basis. Carbon flux measurement turns out to be much more actionable than soil carbon measurements. And the other part on the soil carbon side is that in-field soil spatial heterogeneity is very significant, with 25 to 30% coefficient of variation. And that does effectively require a very large number of samples to take. The other aspect that is really important to understand is that, especially in an agricultural system, ignoring the non-CO2 greenhouse gases provides a false sense of awareness. The real significant share of the greenhouse gases in agriculture are largely methane and N2O. And here, N2O is the real big white elephant in the room, and it has a 20-year greenhouse warming potential of 280. Methane has a 20-year potential of about 80. And I am using the greenhouse warming potential 20, which is for the next 20 years, because that is what matters for us. The 100-year or 500-year greenhouse warming potential is nice. It's nice and informative to some degree. But quite frankly, if we're not mastering the next 10 and 20 years, the next 100 years are going to look very, very different. And it's probably fairly irrelevant to discuss about, how that is really happening for mankind. There is also that I want to point out where we learned is there's a significant year-over-year variability in addition to the spatial variability. So spatial variability is across a field, say 80 acres, we may see 25 to 30%. And year over year, we may also see a 25 to 30% variability of these parameters. And so this variability is making it really complicated and questionable on how we use models that have been developed. And we have seen a number of cases where models such as the DaySense, such as the NDC, and what have you not, and it's reported in the industry, are significantly not representing what is happening in the real world. There are singularities that are happening. There are situations that we have not discovered. And we're finding emissions of methane in places where we would not want to expect them. And we're finding emissions of N2O in a much, much larger amount than what is conventionally being thought of. And that is rather a testament to the limit of the existing database, or the limit of the existing literature that is academically driven, and it is not really driven in a more comprehensive way. The one thing that I would want to point out is that we're very clearly seeing is that cover crops are a very critical factor for carbon balance and for carbon drawdown. I cannot overemphasize the need for cover crops, and ideally cover crops that you grow and then you remove and turn into fossil fuel replacements, essentially. And one point that I'd like to make with respect to spatial heterogeneity and deviation is that of this, the 5% deviation. The California Air Resources Board has a longstanding policy which is derived out of accounting accuracies, where they need to provide data with reasonable assurance. And reasonable assurance means essentially within 5% deviation. If you are deviating and reporting inventory for more than 5%, then it's an adverse material event that needs to be reported. And if we take that thinking forward, then that really informs us as to how accurate and how good our data has to be. If we are having a requirement of meeting a 5% deviation limit from a regulatory perspective, and we have 25% standard deviation across the field, there is a massive mismatch which needs to be certainly aligned with between the regulators and what actually biology can do. This can translate into also differences between measurement methods, where you may have 5 to 10 tons CO2 sequestered per hectare differences, which would translate at current voluntary market prices of $20 to a difference of $100 to $200. Which is a decision point difference for a farmer to do one thing or the other. But if your instrument deviation is that big, there's a fundamental issue here. So with that, that's kind of like the storyline of what I wanted to talk about. We're going to now dive in a series of pictures and so forth. So if you have gotten this message, then I'm very happy, and you can probably go back to your iPhones and iPads and so forth. Although I would like you not to, but I understand people. So we have 6 sites, and they are in California largely, and they are in the South in Arkansas. And that is together with our other teams, which have sites in the Midwest, which are focused on corn and sorghum. And I should mention that the other teams are based at the University of Lincoln, the University of Nebraska in Lincoln. There is one team there. There's a team at UIUC, and then there's a team that is in Oklahoma, Texas, and Florida. So we're covering quite nicely the US. Our site setup is basically pulling together a number of tools in order to measure, and I'd like to run those quickly through. So we have, for instance, net radiometers and PAR sensors that are measuring in and outgoing radiation. We have a rain gauge. We have a open path methane sensor, which is a LightCore 7700. We also have a open path CO2 sensor and water sensor, which is a LightCore 7500. We have a sonic anemometer, which gives us information about three-dimensional flows off the air. We have a camera, so we know what's actually going on the field. We have a direct link with a 4G LTE modem. And we have a very large analyzer for nitrous oxide and CO2. That's a Campbell Scientific provided TGA 200A for N2O and CO2. And we have data processing units on there. Also, I would like to point out that in order to run this this closed path system, you need a vacuum pump that draws a volume through the nozzle here. And that nozzle then that gas goes into the TGA, but the vacuum pump requires about 200 watt continuous. So you can't run this on a solar panel, which means you need to have a power panel. And when you're looking at the distance between this power panel and that power pole, you see that for all our field sites, we had to pull somewhere between 1,500 to 2,500 foot of cable into the ground in order to provide the power. So all to say is this is a very complex, very expensive exercise, and you can't really do that everywhere. But we do have sites, and the sites are typically very close to the field, right? This is the combine, and that gives you the size of the combine and the proximity to a field site. These are actually 3 of our sites and rice fields. One of the things that you start realizing when you actually take measurements continuously, and this is what we do, we don't have chambers there where we measure just once in a while when we get there, is you start seeing phenomena that you really would not see if you would be measuring discontinuously, right? You see, for instance, in particular, also when you take measurements during the cold water season or cold weather in the fallow season, you start seeing that there are actually methane fluxes in these areas happening that you would not monitor if you just focus your eye on looking at the vegetation period. So there's stuff happening in the fields during non-vegetation or fallow seasons. You also see when you do when you do comparisons between various sites that fluxes in certain areas, such as here in California, may be higher than the ones in Arkansas, and it raises the question as to why that is the case. You also see when you do continuous measurements that maybe some of the phenomena, such as in rice, alternate wetting and drying, may be present, but overall, when you start the summation, it may not really be that big. So all of these are consequences of actually measuring continuously. One of the things that we have is we have indeed a site on a peatland or a bog where there is agriculture happening, and what you start seeing from a rice perspective. So rice is one of the topics that I'm particularly interested in, and rice is associated, and I'd like to emphasize, associated with methane production. Rice does not produce methane. It is the soil that is producing the methane. And the soil would be producing the methane regardless of if you're growing rice there or not. So in this peatland, for instance, we have the winter periods, which are, if I can find them, which is over here, where we have high methane production, but we don't really have a drawdown because nothing is growing there. So we're getting data on the CO2 side, but we're also getting on the methane side in the winter periods where there are significant flows. And as we are changing land management practices, such as we are flooding or we're not flooding, we see direct impact of that one. That is essentially a confirmation of basic microbiology or anaerobic digestion in a way. And what we see, and I'm just showing this one here as an example, we see very, very significant seasonal seasonal changes, seasonal variability. So this is a fallow season 21-22. The total greenhouse warming potential equivalence is 62 tons. In 23, we had just 30 tons. And we need to understand why do we have like this 50% change from here to there. Similarly, we had in this vegetation period 14 tons, there 15 tons, and then the last season we had 26 tons in emissions. Right, that is a significant gap, and trying to linearize that one with a model, be it a DNDC or a DASA model, or anything like that, is just not really that fair, because you can make changes and the models are not really capturing that. And there's a number of these examples that we can reference to. We have another site, for instance, where we have a very complex setup in order to look at N2O emissions. N2O emissions, which are associated with fertigation and are associated with irrigation practices in cornfields or corn forage fields. And there we have a setup where we have we actually have 3 different N2O sensors, and we're comparing them, we're testing them, including soil chambers that are actually measuring at the ground level in the fields. And those are very interesting data points that we're generating there. And the irrigation practice is basically fertigation using the cattle manure, which is a recommended practice from UC Davis. One of the things that we started realizing is that if we have this abundance of sensors, we see differences between the sensors. And there are sensors which are so-called open path sensors and so-called closed path sensors. And what we started realizing is the orange sensor here is the open path sensor. The blue one is the closed path sensor. And we're starting to look at carbon fluxes, which is purely the CO2 flux. Just to clarify that, carbon shows up in methane, and there's carbon flux there. But what we're talking about carbon flux here is just purely the CO2 flux. We start seeing that for some reason, and that's an area of active investigation. For some reason, with the very convenient open path sensors, the 7500, so for instance from Lycor, we're seeing not this part here, which we're seeing with the closed path sensor, which is the Campbell Scientific TGA, and that has ramifications. It has ramifications because I'll show that on the later slide. Let me just actually jump to that. It has ramifications because we're over reporting and over measuring in this daytime period, which is really important for photosynthesis. And we have less data in the nighttime period of that, which is where we have the open path sensor just dropping data points. If we're looking at the closed path system, we don't have that. You see a much more even distribution and much more narrow distribution of data points for N2O and for carbon, whereas for methane and for the CO2 in the open path scenario, as much as these sensors are very convenient, we have a problem that we're starting to drop data points, and that is affecting our carbon balance. So when we measure and how we measure really matters. And I'd like to point out another thing. This is comparing of N2O's data and N2O data being the red points, being the blue points, and the red points being the methane data. And what we're finding when we're measuring is that we kind of like have a pattern of where we have in the nighttime a higher flux of methane out that we see here and here. And during the daytime, we have a much lower flux of that, but we have kind of spiking of the N2O going up. So the question then is this actually an issue of physical chemical boundary layers, or is this a biological phenomenon? And that is a topic of current investigation. We're trying to understand, why do we have more data points here? Why do we not have them there? And why do we have them there? And why do we have these spikes here, but we don't have them there? And so this becomes important because you start summing up the data, and when you start summing up the data, then you start realizing that actually the closed path sensor is turning the field into a carbon source. The open path initially turns it into a carbon sink. But when you then start applying a process called gap filling, i.e. you're trying to fill gaps of nighttime data that I showed you later that are missing, then all of a sudden this one here starts moving up to there, and this one started moving up to there. But that leaves you with a question. Now I have like 4 data points, and I'm trying to figure out which one of those ones is actually the right one. Again, this is a topic of current investigation as to why that is the case, but I'd like to really caution that we really need to carefully investigate actually what are we comparing, what are we measuring, and how solid are the measurements. So one aspect of collaboration, this is the zoom up of the various sensors that we have there. There are a couple of interesting configurations here. This one for a time was upside down. And we have these solar-based sensors, and you can see there that they are really deep. That's where they are. That's the solar panel. They're really deep in the cornfield, and this cornfield gets irrigated with water or manure, and this is what we see down there. And so we have these soil sensors there that we have put into the field, and we're measuring actually what's happening at soil basis and as well as in the canopy that is going on there. And we're comparing that one and do calibrations against reference systems such as a Picaro. The blue fat line is the Picaro line here, where the fat orange line is the Picaro line. And the orange line is actually the running average of the agrology sensor. Now, let's talk about cost here. So the Picaro, which is like this system, is running between $50,000 to $80,000. And then you need to multiplex this. So you're looking easily at $120,000 with a couple of soil chambers that you might have there, which are coming in at a pop of $10,000. So that's not really that conducive for individuals to purchase that. And the hope is that if we can come up with sensors that are much more cheap, much cheaper and equally accurate, we should be able to actually generate more data points. And I'd like to have you think about the purple air paradigm for that matter. You know, when air, and we do have a purple air sensor here, actually, that's somewhere there. The purple air came out, and we have like $300 devices that people can purchase. It is just all of a sudden spurring a revolution because people start going out buying their sensors, and you get a data set, and you see immediately how good or how bad is the air in your neighborhood. That is real-time data delivery, and that is real-time giving people the power to show to people in power as to what is going on with the air. And all of a sudden, there's a focus on air pollution, and there is actionable data, and there are actions that are being taken upon that one. People in the Bay Area had purple airs everywhere, and when the fires and the smokes were going on, started all of a sudden seeing people taking actions and demanding actions on schools for kids to be inside and outside and so forth, and it became a big deal. But it is all driven not because there is one air sensor for the entire county, which is provided by the EPA, but because we have hundreds and thousands of sensors which are purchased by private citizens that are contributing to the private citizen size. And that is only enabled by way of having actually a large amount of abundant calibrated sensors in the field. I'd like to dive into one topic, and soil carbon sequestration is a big deal, or was at least a big deal. There are certain regions which are kind of like moving away from that, such as Europe, because they start realizing that there's a serious problem with spatial heterogeneity. So this is a typical grid layout, how we sampled on all our sites. And I have, over the past 3 years, pulled about 3,500 soil samples, soil cores, analyzed them, and we put them into data and all that. And we analyzed them across, pretty much all parameters that we could look at, or that are relevant and that are, making sense, ammonia, nitrate, nitrogen, soil texture, organic matter, and total carbon. And what we started realizing is that, and this is not really that novel because people should actually know that in soil sciences since about 20, 30 years. This whole concept of soil carbon sequestration, I'd like to mention, comes out of the Kyoto Protocol. The Kyoto Protocol, which was never ratified by the United States, that's why it was never a problem or never a topic in the United States. The Kyoto Protocol suggested a soil carbon sequestration along the line of the 4 per mil initiative. The problem with that is people fairly quickly realize that if you take a large number of samples, you start realizing you have a standard deviation of like 20%, 30%, depending on what parameter you're looking at. But for carbon in particular, it's like 30% is like your standard deviation de rigueur. And you have this, regardless of if you're taking 8 or 10 samples around this small little spot here, or you're taking it across the entire field. So if you start comparing the standard deviation of 28% here to the 25%, which is like for the entire field, you start realizing that a field that is per soil map, a very homogeneous field, has actually a lot of heterogeneity across all depth and across the entire field. And we have seen this in all fields, and it's not really that novel. Other people have seen similar. They're a little bit shy sometimes about reporting that, but it's a fact, it's a reality. And that makes it very difficult because statistically, you then, especially when you look at this 5% paradigm that I mentioned before, that the regulatory body or the accounting body wants to see, and this is very problematic because it means that you have to look at very long time horizons in order to actually demonstrate and very large sample numbers if you want to demonstrate that actually something happened. And you're looking at 300 to 1,000 samples in order to fix that, or you're looking at, here we calculated out how many samples we needed, 970, I think, in that case. And that is the standard deviation across the whole thing. And in fact, actually, if you're going even further in the statistical analysis of that one, you start realizing that, you need to use very sophisticated and very advanced and stilted statistical analyses on this one. And you will start realizing that you're not really meeting, things such as normalcy of the samples, et cetera, et cetera. You're just violating so many statistical assumptions here. And so here, for instance, is the number of samples that we would need to take in the various individual fields, right? You're looking at 300 to 1,000 samples. This has been pointed out back in '99 in a paper by Gartner and Volschläger. So it's not really novel that we're finding this, but here we are. And I guess with that one, I'm coming to the to the Q&A section at this point. This is just what I wanted to share. We have about 30 minutes, which is fantastic because that should make for a very lively conversation.
Thank you, Dr. Michael. I really appreciate the time you dove into this and the deep depth you took into this pretty technical area. So I think the first question I'm going to have for you is one that we've had several people ask us over time, but I think a lot of people may also kind of wonder about it. It's going to be a little less deep in the technical end of things, and that is, can you just walk kind of high level over how gas flux measurements can quantify soil carbon sequestration? And this is a question we get because this is a relatively advanced technology compared to, say, soil sampling, other approaches or modeling. And so getting a sense of, the methodology that you do to understand carbon sequestration via gas flux would be really great.
Yeah, let me, okay. That one. So what we have here is we have one sensor here, for instance, or this one here, which would be measuring N2O. And this one also measures CO2. We have another sensor there that is measuring methane. And that's a laser-based system. And we have another sensor here that is also measuring CO2, which is an infrared sensor. And they are basically measuring the concentration of the gas at that particular moment in time as it's passing either through the inlet nozzle or through this open path here. This is why this is called an open path sensor. I don't know if we have a better sensor here. So there's a path of the wind goes through there. The wind comes from about this point, goes through there, carries a molecule, and then that molecule is affecting the spectroscopical image. And we then see there's a reduction in the spectroscopical response. And because of that, we're then determining that is the concentration of that element at that point in time. And this is happening at a frequency of 10 hertz, which means 10 measurements per second. And these data points are being accumulated, and then that is correlated with a three-dimensional wind flow. And what you then do is, through some fancy math and differential equations, you strip everything away that is essentially linear, and you just take the stuff that is moving up and the stuff that is moving down. And every time there's moving something up and you have a concentration, you add that one up. And every time there's moving down and you have that concentration, you add that one up. That's very simplified. And then you basically calculate out what is the balance, right? Is there more stuff moving down or more stuff moving up? And then that tells you if you have like a flux going in or going out into the flat plane, right? The flat plane being this one here. And then we have the value for CO2, the value for methane, and value for N2O or any other gas that you would have a sensor for. And we add that whole thing up. which is something along the line here. So we integrate that. So we take here, for instance, we would be taking all the measurements, which would be fluxes. You can see the thin green line actually is, in this case, that's our N2O flux. And I didn't really went into that one, but this is when there's an irrigation event, there's an irrigation event, there's a fertilization event, there's a fertilization event. And as a response to that one, you see that the flux, which means the amount of moles on a per square meter basis per second, is spiking up. And then what we do is we just simply integrate that over the entire season, which goes from here to there. And then we report that one as our flux for the season. And that's that. And then we multiply that by your favorite greenhouse warming potential. And as I said before, I use the 20-year one because that's the one that matters from a climate change perspective. And that gives me that balance. And we would have to do that for separately for our CO2. We do that for methane, and we do that for N2O. Just because this is essentially unpublished data, I'm just not going to show you the methane and the N2O data because we're still trying to evaluate that data. That somehow answer the question.
Yeah, no, thank you, Dr. Michael. That helps a lot. So essentially, you just kind of look at it's a budget. You look what's going in, what's going out, and you track the changes over time.
Yes.
And it looks like the sensitivity here is really, really high to changes. So we have another question coming in. I see, and I'm going to start catching up here from Evan Hazlett. So his question is, if you need that many samples for a reasonable level of confidence, does that suggest that soil sampling is not an economically effective means of measuring carbon, or that soil carbon is not the metric that we should be using?
Yeah, I think you could say that.
Okay. Excellent. Well,
that's a short answer on that one. Yes. No, soil carbon, if you really would want to monitor soil carbon sequestration, you have to do that over long periods. Now, am I saying that we shouldn't do those measurements? No, I'm not. And in fact, I would hope that there are finances available that we can actually go to these fields in 5-year term and we can measure again what was happening. But the reality is that you really need to go back there. And I do have to say that in our fields where we took 80 samples per acre, 80 samples per 80 acres, so 1 sample per acre, we are by any statistical analysis, we're statistically underpowered. The approach that we're taking would not fly at all statistically if you'd go into life sciences or biopharmaceuticals or medical sciences. And I have worked in that area for 20, 25 years. Statistically, we would not, the approach would not fly. You would be required to take 300 to 1,000 samples, and that's it. And why do we do that in medical sciences? Why do we not do that in ag sciences? Well, first of all, medical sciences pays much more for any end product. And on the other side, we have regulatory bodies that are overlooking that, the FDA. And it is encoded that you got to have your statistical significance. Our approach is here, our statistical, if you apply those common statistical methods on that one, we would not survive. And it has been written about. I'm not the first one. I'm just regurgitating what other people have already done, and pointed out before. Yes, it's a serious problem, and that is also the issue when you overlay these long time horizons of 7 years or 10 years that we would have to go back in order to verify if our practices are actually having a benefit. That really should change and should impact the approach that we're taking in this very urgent time to change our approach to carbon emissions. We need to have methods which are giving much more real-time feedback as to what we are doing. And I can, I'm showing you 2 seasons of data. This is 1 year of data. And I can show you very specifically what is happening here with the N2O emissions, for instance. I can have a conversation with a farmer on this one and say, hey, we need to change the practice here, there, and everywhere. And farmers are the ones, they are very responsive to what the market wants. The farmer is delivering what the market wants. And if we have a whole bunch of omnivores that are wanting to eat milk and cheese, then that is what the farmers are producing. It is me as a consumer who's driving that. It is not the farmer who's like just doing it. And so similarly here, for instance, we had conversations with this farmer here, and we said, look, this is a real problem, your winter emissions with methane. And what can we do about it, right? Can we like not flood the field and therefore reduce the methane emissions? And that's a topic of our current investigation. So yes, we can have impact, but we need to have tools which are much, much more near term.
Got it. Excellent. So thank you for that. I have another question here coming in from Sue Patrick. Was there any relationship between your results for CO2, N2O, and CH4 and the amount of water vapor at the site?
So that's a deep question in one way, because water vapor does actually play into the whole thing. There are two measures. You can talk either about the concentration, which would be including water vapor, and there is the aspect of the mixing ratio, which is a parameter that is without water vapor. And between the Flux networks in Europe as well as in the US, there seems to be a little bit of a difference who wants to report what. We are calculating both, or we're measuring both, and it depends a little bit on the sensor as to what you're measuring. Our close pass sensor has a Nafion dryer before, so by definition, the gas that goes in there is actually a dry mixing ratio gas. So it's a dry gas without the water vapor. We're measuring the water vapor separately with a 7500. And that one gives us that number. And then the open pass sensors, by definition, because they are measuring the air as it comes through, are measuring the wet gas. And so, yes, water vapor plays in there, and it does have it plays a role, and it needs to be calculated in or calculated out depending on which parameter you're using.
Thank you. All right, so we have another question coming in here from Jason Bradley, who wants to know if we'd be better off using measuring soil carbon using technologies like inelastic neutron scattering to achieve or overcome the 300 to 1,000 samples per field for statistical validity. And so I'll caveat this with, I am not sure about Dr. Michael's familiarity with inelastic neutron scattering. I'm glad this question isn't coming to me because I don't have familiarity with it. But if that's something within your wheelhouse, we'd love to hear an answer.
So I'm, I do have a radiology permit, and I use radiological tools in my lab, not for that particular part, but I know that, and I think this is what it's referring to, that Lawrence Berkeley National Lab has actually a team that is actually actively working on using these technologies and measuring, essentially using these tools in order to measure carbon emissions, solid carbon sequestration and solid carbon content in the soil. Yes, anything along that line that you could basically put behind a tractor, you could drive across the field and you can take a measurement of that one, would be fantastic. The problem, and we glanced over that a little bit, is that it does not really change the fact that you still have heterogeneity across the field, and that the carbon uptake in comparison to the carbon that is in there and in comparison to the variability across the field is relatively low. So the issue on soil organic carbon sequestration is not only one of the is not one specifically of the variance. It is one of the variance in comparison, in conjunction with the relative slow change over time. And so it will take you quite a while to move your data point out of this error bar that you have when you take a measurement. The error bar does not go away just because you change the method. The error bar is what the spatial variability is in the field. What you need to do is you need to move out of that one, and that will take such a long time. That's why we have the 7 to 10 year horizons where you only will be able to see if you really had an impact.
Great, thank you. We have another question coming a second ago from Padma. Are there any effective portable methane and N2O sensors that can be just added onto greenhouse gas chambers? We're trying to explore how to reduce costs by reducing automatic greenhouse gas chambers, since we have multiple locations for measurements.
Well, I would just probably refer that question to the sales department at agrology.
Yeah, I was about to say, as I read this one out live, it's going to come back to me. And so, what I'd emphasize here real quick is that we've made an enormous amount of progress working with Dr. Michael to cross-validate the nitrous sensitivity of our chambers this year, and we're really excited about that. We are working on methane. It's a slightly trickier gas to detect at an affordable level, but we're hoping to have a clear path to that capability towards the end of this year. And of course, with the way our business model works, if somebody becomes a customer now and is really looking at nitrous and carbon today, when methane capability becomes available, now their systems will be upgraded without a cost. So we'd be happy to talk more with you about that, Padma, and I can assure you that our devices would reduce costs and reduce labor and just make things a lot easier.
It's
Then
a
next up,
let me maybe hook into that one. So
yep.
To understand where the challenge here is that the concentration of methane that we're looking to measure is about 2 ppm, and we're trying to do that with an accuracy of about 2 ppbs or something on that range, right? With a precision of 2 ppbs, that's what I should say. The concentration of nitrous oxide is typically in the range of about 0.3 ppm or 320, 340 ppbs. And we're trying to get that one also down to about 1 ppb precision in there, right? So that is the challenge, right? We're not measuring very high concentrations. And for comparison, CO2, for instance, in the atmosphere is somewhere in the range of about 400, 420 on the baseline. And we're measuring up to about 800 ppm if we have these events of where there's a lot of CO2 coming out. And so that is the challenge. And as you're measuring something at a very, very low concentration, your cost just exponentially goes up. And especially if you want high precision and high accuracy, the cost just goes through the roof. And that is what is explaining the cost in here. And that's just the nature of that. If you're measuring methane in a biogas scenario, for instance, where your methane concentration is 50%, CO2 is 50%, your sensor, at a high precision is going to be like $5,000 to $7,000, right, at the most, right? If you're going in, you're trying to measure 2 ppm, well, things are getting more pricey.
Yeah. Thank you. So next up, we have from Stephanie Kortman. First of all, hi, Stephanie. Nice to talk to you again. The question is, how does including the OpenPath Flux data affect daily and seasonal emission estimates? Are those estimates lower when including those data? Are the non-daytime measurements proportional to the daytime closed path measurements for any one crop or treatment?
That's a compounded question. That's complex. Normally, I ask these questions. So let me unpack that a little bit. First of all, as I said, it's an active topic of research, right? Because in CO2, you obviously have the complexity that on what I want to show here, you have the complexity that this is your nighttime activity, and your nighttime activity is purely respiration. Your daytime activity for plants out on a field is both respiration as well as GPP or photosynthetic activity and productivity. So there is a complexity in here. And in our models, I would say, and our understanding is still a little bit emerging, I would say, because we started on occasion also measuring negative fluxes in the night times. And normally you would say that should not be the case because there is no photosynthetic activity. But there may be other boundary layer conditions that may affect that, which may be purely physical chemical. But we may also have a phenomena where we actually have carbon sequestration during night. I cannot really fully exclude that. I'm not. I want the data to drive us here. And so yes, that does affect how we look at things. If we in a model and then in the gap filling process or overemphasize this green area, which is like the photosynthetic activity, we will report a significant higher a significant lower carbon intensity or higher photosynthetic activity than that is actually there. And by way of having more data points here and much less data points there, and the difference between them can be about 60%, and you can go across all CO2 sensors out there, and you will find the same pattern. I've done that. Then you will see that there is a problem. And if we then go systematically about it and we apply a gap filling process to the whole thing, we fill these gaps here, then the question is, how accurate are we filling a lower pool of data with data that we pull from a lower pool of data? How much bias are we introducing? And it's a huge problem. It's a systematic problem. We need to address that because yes, indeed, it does. We may be over-reporting carbon sequestration. That may be the consequence. Yes.
All right. Thank you. I think we have a follow-up here. What about for N2O? Doesn't look as different across 24 hours.
No, it doesn't. And also in N2O, and I'd like to point that out. This is our closed path and our closed path sensor. So we have a double measurement here for closed path CO2, and the difference here between an average is like in the single digit percentages. So the error we're using the closed path data versus the open path data. On first thought, to me, would be probably significantly lower. And for N2O, yes, I mean, this is somewhat misleading because the number of difference in data points, between the highest and the lowest in this one here, and this side, for instance, is very close together, right? And that's a difference of like 5% of data points, and that we could like there versus here where we have like between 20 and 40% data point differences. So this is a problem that's really specific to open path and very specific to CO2.
All right. Thank you. So at this point, I think we might have one more question here, which, what do you think about, broader applications for affordable gas flux measurement in agriculture, and what kind of future do you see there?
Well, so the two key greenhouse gases out of the agriculture is methane and N2O, right? N2O is associated with fertilization, and N2O fertilization, we need to understand that all nitrogen that is ending up on a field, one way or the other, even if it's organic fertilizer, is a fertilizer that was produced from fossil fuels. Haber-Bosch produces and gives us the ammonia, and the ammonia gets converted into whatever, runs into a plant, becomes a protein, is being fed to cattle, and eventually gets pooped out, and then that gets distributed on the field. And so even if it's organic fertilizer or synthetic fertilizer, all nitrogen is essentially a fossil fuel. And we got to be very clear and aware of that one. So if we do want to reduce our fossil fuel dependency, our fossil fuel input, then addressing nitrogen as a fertilizer is definitely a key important aspect for agriculture. The other aspect is methane, and methane is on one side the enteric fermentation, which having worked in anaerobic digestion with methanogens since 87, I would, and to just expand on that, methanogens are archaebacteria. They're here since the Big Bang, and I do laud everybody's excitement of being able to trick around an organism that has been around for billions of years on this planet. Good luck, and the archaea is probably going to say, "Hold my beer." And the other aspect as to where methane is coming from is indeed out of the soils, and specifically the rice fields. So rice fields are the second largest methane emitter after the entire animal complex of enteric fermentation and manure management with respect to methane emissions. And again, it is the rice field, it's the soil, and it's the anaerobic conditions that are on the fields. It is not the rice plant per se. Rice does not have an ability to produce methane, the rice plant. It is the soil that is in the soil microbes, the archaea, that are producing the methane. And that is something that needs to be addressed or should be addressed one way or the other. But quite frankly, I'm the most hopeful way of addressing that one is by way of crop residue management, i.e. removal of the crop residues from the fields, so that you're not providing more feedstock for methanogenesis. That has been researched for many years, and it should be one of the primary practices in rice fields. There are some ways of how you could reduce methane emissions, and the farmers in Arkansas, one particular, has shown us, how we can do that. So shout out to those people in southern Arkansas. But again, there's more work to be done on that one. So from a perspective there, I think monitoring and seeing what practices at field scale and field testing level pan out is where we see that in probably the next 3 to 4 years, I would say. But as I said, we can get results if we have measurement stations on fields within a season. If we do that statistically well enough with those sensors. The smaller chambers that agrology is working on, I see as a way of expanding into broad application and monitoring and confirming how the practices actually work at scale basis. Farmers are putting weather stations out and they're measuring everything that is going on with that one. Why not have a measurement station out there that tells them how their daily practice, how their monthly practice is affecting the emissions on the field?
All right. Thank you. I think we have time for one last question here, given that these questions tend to be pretty complicated. So last I have here from Tim, given variability of N2O flux measurements from spatial, year-over-year, and sensor precision, how many season crop replications are needed to come up with a good number for a so far unstudied crop or region? Relatedly, does each crop need its own studies, or will similar crop, let's say brassicas, have similar N2O fluxes and so can be aggregated?
So let me put that into the bigger perspective. We're running 6 towers, and there is probably, at most 20 towers in the US that actually have the capabilities that we have, including methane and N2O measurements. There may be 20 to 30 towers globally that have these capabilities. N2O measurements are very complicated, and people have tried it, and they have not really been that successful. Where we're, and RPE, the funding agency that funds us, is doing far out, high risk, high complex, and expensive research. And there's a reason why they engage in this one, because it is far out and it is complex and it's complicated and it's a cutting edge of what we can do. So we'd like to reduce this to practice very, very quickly. But I really would caution, and because of the limitations of the Department of Energy having to focus on bioenergy crops, this is not a program of the USDA yet. We wish, and we pointed out to the USDA, that this needs to be done at scale at the US Department of Agriculture. And we would have hoped that the Climate Smart USDA program would have done much, much more measurement. That is not yet the case. And there is an effort at the USDA underway to pour, I think, about 600 million into greenhouse gas measurements at agricultural level. That has to come from and needs to be supported and run out of the US Department of Agriculture. There is a massive amount of need for these sensors to understand that. And that's a long-winded answer to say that we don't really have good measurements for brassicas. We have measurements for, the other teams have it for, we have for corn and for rice. Other teams have it for corn and sorghum, but that still has to be published. We have a couple of really excellent teams in other countries. Shout out to the team in Ontario and Guelph around Claudia Wagner-Riddle that is doing a lot of research on organic fertilization of pastures and cornfields. There's a team in Waikato in New Zealand that does a lot of work on pasture management. But there are teams in Europe that have done that, and they have decided that after a year or so, they couldn't continue. We do not have this large database of that we should have in order to understand what's going on. And on the other side, yes, we'd love to deploy this on other fields, other crops and so forth. And to answer the question about how many replications do we need, I think if you'd be running a 3-year trial, you probably are getting a sense of what your variation is. If you'd be running a 24/7 agriculture, such as in our forage fields, you probably can get away with 2 years of data because you have, replications and so forth. But the scientific standard is typically, let's do 3 replications through 3 seasons, 3 years. If we have the money, we can put the sensors out, we can measure it. I think that's a very reasonable approach. And if you do it, you can scale this, right? We can run fields side by side by side by side. Very quickly, we would be getting that database. Just takes money.
All right. And we are at 2:00. If anybody else has a question, they're welcome to pop it in the comments. We can try and follow up by email. You're also welcome just to reach out to anyone on the agrology team. We can pass questions along. I want to thank everybody for attending today and for asking questions appropriate in detail to the caliber of the presentation and the presenter. That was fantastic. It was great hearing all of this and learning about how the power of gas flux to understand climate positive agriculture and to help farmers really see the power that they have in their hands to change their practices for the benefit of the climate. So thanks everyone for coming. And for those who want more information on agrology, we'll be happy to follow up. Oh, and thank you, Dr. Michael. Excuse me, I should have led with that one right there. We really appreciate all your time
Thank
today.
you, everybody, for
And
bearing with me, and apologies for like not doing it last week, where my voice was non-existing, and today it's somewhat existing.
Yeah, no, it happens. It happens. We'll make sure to get this out, Dr. Michael, to everybody.